So today we're going to talk about meiosis, and meiosis is the process by which we take a diploid cell, which contains 46 chromosomes, and we cut the chromosome number in half through two cell divisions, meiosis I and meiosis II, to get four genetically different haploid cells, and those cells contain 23 chromosomes each. So we're going to talk a little bit more about chromosomes and chromosomal structure, and then we're going to take a closer look at how the cell goes about cutting the chromosome number in half. So let's get started. So when a cell is not dividing, the DNA is loosely coiled in the nucleus in something that's called chromatin. So think of a pot of spaghetti. After it's been cooked, all the little spaghetti strings are, loosely coiled in the pot. You can't really differentiate one string of spaghetti from another. That's the way DNA typically looks in the nucleus when the cell is not dividing. When a cell is preparing to divide, the DNA is tightly coiled into structures called chromosomes. So let's just say we took that, cooked pasta and we were able to sort of go backwards and dry it out so that it's like, stick straight, like, and ready to be put into the box. That's a chromosome, or that's what DNA will look like when it's in chromosomal form and it's preparing to divide. It will tightly coil about itself so that it's just this one kind of stick-like structure that can be moved and manipulated wherever the cell wants it to go. Now, chromosomes before each cell division need to be copied. So that is called DNA replication, and we talked about how DNA replication is semi-conservative, and the base pairing rules ensures that our DNA molecules are identical to each other. So after DNA replication has taken place, each chromosome consists of two identical sister chromatids, and each pair of chromatids is attached at the center called a centromere. And when a cell divides, each new cell will get one chromatid. So if we take a look at this diagram here, up here we have, before DNA replication, we have a DNA molecule, and that one DNA molecule is called a chromosome. Once it has been replicated, each DNA molecule, one here and one here, are called a chromatid, and this whole structure is called a chromosome. So chromosome can refer to either an unduplicated DNA molecule or to a duplicated DNA molecule. This diagram back here is pointing to the sister chromatids, one along here and one along here, and then again they're attached at the center called a centromere. You should know the structure of a duplicated chromosome and where a centromere is located. So the chromosomal theory of inheritance states that genes are passed on from parent to offspring by means of chromosomes, which makes sense because we talked about how Griffith, Avery, and the Hershey-Chase experiment confirmed that DNA was a molecule of heredity. So on DNA, there are sections of DNA called genes, and those genes code for proteins, and those genes are passed on along the DNA molecule, which is why you look similar to your parents or similar to your brothers and sisters. Now, each organism has its own characteristic number of chromosomes. So if we take a look at the diagram here, penicillin, which is a bacterium, or a fungus, I should say, has anywhere between 1 and 4 chromosomes. Humans, as we know, have 46 chromosomes in their diploid cell. Dogs have 78 chromosomes. So each organism has its own distinct number of chromosomes. That's what I want you to take away from this, is that each different organism has its own distinct number of chromosomes, and on those chromosomes is the genetic information that can be passed on to the next generation. So there are different types of chromosomes. Specifically when we're talking about humans, there are sex chromosomes. There's only 2 of them, and there are autosomes. Now, the sex chromosomes are called sex chromosomes because they determine the sex of an organism, and they are the last pair of chromosomes when we look at a karyotype, and we'll be looking at those a little bit later on. Males have an X and a Y, And if you are a male, you inherited your Y from your father. If you are a female, you inherited an X from your father. So fathers can pass on either X or Y chromosomes to their offspring. They only pass on one of them. Okay. Mothers or females have two X chromosomes, and if you're a male, you received your X chromosome from your mother, and if you are a female, you received another X chromosome from your mother. So the sex chromosomes help determine sex. All the other chromosomes, all other 44 chromosomes, are called autosomes, and there's 2 copies of each autosome, 1 copy from each parent. So chromosome number 1, you have 2 copies of it. 1 came from mom and 1 came from dad. So in total, we have a total of 46 chromosomes in a diploid cell. 44 of those chromosomes are autosomes, and the remaining 2 are the sex chromosomes that determine sex. So haploid versus diploid. You need to know the difference between haploid versus diploid. This is important. Haploid cells are cells having only one set of chromosomes and therefore only one set of genes. So sperm and egg cells are perfect examples of haploid cells. Those are the reproductive cells. Those are gametes. And haploid cells are produced by the process of meiosis, which we're going to learn about in a little bit. So diploid cell is cells containing or cells having 2 sets of chromosomes. Those cells are your somatic cells or your body cells, and they contain chromosomes from both your mom and your dad. So if we take a look at this diagram here, I love this diagram. We've got our diploid organism. So your female diploid organism, your male diploid organism. And both males and females can go undergo meiosis in order to produce haploid cells. So 2n just represents, the number n represents any number. It could be 4, it could be 6, it could be 23. And then if it was 23, 23 times 2 is 46. So we know that this cell is a diploid cell. A diploid cell undergoes meiosis and produces haploid cells or N cells. So this would be 23. Meiosis produces our gametes or our haploid cells. And haploid cells undergo fertilization. So a sperm and an egg cell, when they meet and fuse together, that's called fertilization. And after fertilization, we then start out with the first cell of a new organism, and that's called a zygote, and that's a diploid cell. So this kind of shows the diploid life cycle of yourself or an organism. So we have our zygote. That zygote undergoes mitosis, and you get your full diploid individual. And then a diploid cell will undergo meiosis to produce haploid cells, and then those haploid cells can then go and make a new diploid organism. So let's take a closer look at meiosis. So meiosis results in new cells with half the number of chromosomes as the original cell. It produces what we call gametes, or your haploid reproductive cells. In males, those are going to be your sperm cells. In females, those are going to be your egg cells. Cells preparing to divide by meiosis undergo 2 parts to it. They undergo interphase and they undergo cell division. And there's 2 main cell divisions in meiosis, Meiosis I and Meiosis II. As I said before, scientists were really boring. We name things after what they do or what their function is. In this case, we're going to name things after what they do and in order of what they do. So Meiosis I comes before Meiosis II. Let's take a closer look at interphase. So interphase is the period of the cell cycle between cell divisions. So it's sort of like a prep phase for the cell before it actually goes through and divides. So interphase is divided into 3 parts. G1, it stands for growth 1, and it's where the cell grows and increases its supply of organelles and proteins. After the cell has gone through G1, it will then go through S, and that S stands for synthesis, and this is when DNA replication is taking place. Next, after DNA has been replicated, the cell finalizes its growth and supply of protein, so that's G2, that stands for growth 2. And then Only a handful of cells are ever in G0, and G0 is just a phase where the cell is not preparing to divide at all. So cells that are usually in G0 are brain cells. Those neurons in your brain are often in G0, which is why it's such a horrible thing to get a brain injury, because your cells can't divide and replace themselves because they are in G0. They're not preparing to divide anymore. So some vocabulary that we need to know before we move into the stages of Meiosis I and Meiosis II. First is homologous chromosomes. Homologous chromosomes are chromosomes that carry the same genes, but one comes from mom and one comes from dad. So think of socks. Socks come as a pair. Imagine one of those socks coming from mom and the other sock coming from dad, and that's your pair of chromosomes. And they have the same genes on it, just like if you had socks, they would have, your pair of socks would, each sock would have the same pattern on it, let's say dolphins or something. When your cells in meiosis are preparing to divide, we're going to end up forming a structure called a tetrad. And right there in that diagram, you see this structure called a tetrad. And what a tetrad is it's just a structure that contains the 4 chromatids, and then one chromosome comes from mom. Here it's showing in red, and the other chromosome is in blue, and that's the paternal chromosome. So a tetrad is important because this is going to help us ensure that genetic variation takes place. We want our gametes at the end of meiosis to be genetically different from one another, or as genetically different from one another as possible. So genetic variation, how do we make sure that cells at the end of meiosis are genetically different from one another? The first way we do this is a process called crossing over. So crossing over is when homologous chromosomes pair up and they exchange portions of their chromatids. So this occurs during meiosis I of prophase. You are going to need to know the 2 things that ensure genetic variation in meiosis. The first one is crossing over. You also need to know when it takes place during meiosis, and it occurs during meiosis I, and it occurs during prophase I of meiosis I. So here we've got our homologous chromosomes. The brownish golden one will come from, is the paternal chromosome, comes from dad. The pink one comes from mom. And you can see here that they have the same set of genes on them, okay, A, B, C, D, E. A, B, C, D, E. The only thing that is different is their capitalizations. So the capital A might code for brown eyes, but the lowercase a might code for blue eyes. So, but A represents the gene for eye color. So they have the same order of the genes. That's why they're homologous chromosomes, but they can have different versions of the genes on each different chromosome. And here we see that the maternal and paternal chromosome are exchanging genetic information. And with the exchange of genetic information, we now have a composite chromosome that allows us to kind of increase our genetic variation. The other way we increase genetic variation is through independent assortment. So independent assortment is when chromosomes segregate independently, and this occurs during meiosis I of metaphase I. So here's an overview of meiosis. We are taking, up here, we are taking a diploid cell right here, and we are going to undergo meiosis. This is showing metaphase I in meiosis I, and we can see that in red, all the maternal chromosomes are on the right-hand side of the cell, and all the paternal chromosomes are on the left-hand side of the cell, but they don't have to line up that way. They could line up so you get one paternal and one maternal on the left-hand side, or one maternal and one paternal on the right-hand side. So this right here, showing in meiosis I metaphase 1 of meiosis 1, this is showing up the two different ways that the chromosomes can line up during meiosis 1. And if they line up differently, and this is completely random the way they line up, if they line up differently, we get different combinations or a different combination of gametes in at our end product. So here we've got both paternal chromosomes in two gamete cells. Here we have two maternal chromosomes in two gamete cells. But here in all of these, we've got one maternal, one paternal chromosome in our gamete cells. So we've got increased genetic variation over on this side as opposed to this side. So imagine doing this with 46 chromosomes. You're going to get a lot of genetic variation, especially if the chromosomes can line up randomly during metaphase 1. So going through meiosis 1, these are the steps that the cell will go through during cell division. So prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. What I would like you to do now is pause the video, jot down these notes, and then I'm going to explain these on the next slide. Oops. So meiosis 1, we will have gone through interphase, and remember that interphase consists of G1, which is a growth cycle, S, which is synthesis, is when DNA replication is taking place, and G2, kind of finalizing our growth. So our cell will start out as a diploid, so this will be a diploid cell we're going to start out with in meiosis. So we've got to start out with a diploid cell, and we'll go through interphase. We'll prepare to divide. Then we'll go through meiosis 1. And here we've got our 4 chromosomes. We've got 4 chromosomes. The homologous chromosomes are going to pair up. Remember, one of these is coming from mom, the other one is coming from dad. One is coming from mom, the other is coming from dad. So the homologous chromosomes are going to pair up. The spindle fibers are beginning to form, and they are going to eventually attach to the centromeres of each different chromosome. During prophase 1, this is when one of our types of genetic variation can take place, and this is when crossing over. This is when crossing over takes place. And crossing over takes place when we form this structure right here, and this structure right here again is called a tetrad. Okay. Now in metaphase 1, the homologous chromosomes are going to line up in the midline or the equator of the cell. This is referred to, this midline, this kind of imaginary midline in the center of the cell, is referred to as the metaphase plate. And this is where our second part of genetic variation takes place. This is where independent assortment takes place. So how the chromosomes line up along the midline is completely random, and then in anaphase, these chromosomes will get pulled. They're going to be pulled this way towards the poles of the cell. So the spindle fibers are going to contract, and they're going to pull those chromosomes towards the opposite poles of the cell away from each other. In telophase, the nuclear membrane is going to begin to reform, the spindle fibers break down. In cytokinesis, what cytokinesis is the division of the cytoplasm. So we're actually going to divide the cytoplasm in half, and the end result of meiosis 1 is that we're going to produce 2 genetically different haploid cells. So you need to know the outcome of meiosis 1. You need to know what starts meiosis 1, which is a diploid cell, and we are going to then end up at the end of meiosis 1 with 2 genetically different haploid cells. Then we're going to roll straight into meiosis 2. Now, this is important. Neither cell goes through a round of chromosomal replication before entering meiosis 2. There is no interphase between meiosis 1 and meiosis 2. That's important. So no interphase. We're going to just go right into PMAT, prophase, metaphase, anaphase, telophase, and divide again. Now pause the video here. I want you to write down what happens in each phase of prophase 2, metaphase II, anaphase II, telophase and cytokinesis II, and I'm going to explain what happens on the next slide. So meiosis II. We are not going to go through another round of interphase. So we're not going to replicate our DNA again. That's important. So we're going to go straight from, taking a look over here, we're going to go straight from meiosis I. Right here, we've got our two genetically different haploid cells, and we're going to go right into another round of cell division. So in prophase II, our chromosomes are here, our spindle fiber is going to begin to reform, our nuclear membrane has broken down, and this is occurring in both cells. In metaphase I, our chromosomes, so the spindle fibers will attach to the centromeres of our chromosomes, and they will align the chromosomes along the midline or the metaphase plate. We've got them lining up along the middle of the cell, that metaphase plate. The spindle fibers will then contract and pull each chromatid, now we're dealing with chromatids, pull each chromatid towards the pole of the cell. So we've got the spindle fibers contracting, and each chromatid is being pulled toward the opposite pole of the cell. And then in telophase II, we've got the spindle fiber breaking down completely, the nuclear membrane beginning to reform around those chromosomes, and then we've got, of course, cytokinesis occurring. So that's the division of the cytoplasm. So at the end of meiosis II, we will end up with 4 genetically different haploid cells. Now, gamete formation. How we produce our gametes occurs differently whether or not you are male or female. So in male animals, meiosis will result in 4 equal-sized gametes called sperm. This process is called spermatogenesis, and it occurs in the testes. So here in this diagram, we're taking a look at what happens in meiosis I. We're going to start out with our diploid cell. That diploid cell is going to undergo meiosis I, and we're going to get 2 genetically different haploid cells. Then those 2 cells are going to undergo meiosis II, and we're going to result in 4 genetically different haploid cells, and those cells are going to end up producing 4 sperm cells. In females, this occurs a little bit differently. So in female animals, only 1 egg cell results from meiosis. The other 3 cells are what we refer to as polar bodies, and they are discarded or broken down, and they aren't used. This process is called oogenesis, and it occurs in the ovaries of females. So again, we're going to start out with a diploid cell, and that diploid cell is going to undergo meiosis I. We're going to end up with 2 genetically different haploid cells, but you're going to notice a difference between our haploid cells here. One cell is much larger than the other cell, and the reason for that is that there is an unequal cytokinesis that takes place. There's an unequal distribution of the cytoplasm, so that the majority of the cytoplasm ends up in one cell, and then the other cell ends up with only a tiny bit of cytoplasm and a couple of, and their genetic information. Both of these cells are going to undergo meiosis II. Again, there's going to be an unequal division of the cytoplasm with this cell, so that we end up with 1 cell that has all of the cytoplasm and 3 cells that have hardly any cytoplasm. All they really are made up of is nucleus and genetic information. Those cells are called polar bodies, and those are broken down by your body. Those are not used for reproduction. And then we've got our cell with all of the cytoplasm and its genetic information that will end up resulting in our egg cell. So just to overview meiosis, okay, meiosis produces 4 genetically different haploid cells. Meiosis is used for reproduction in eukaryotic organisms, or organisms that have multicellular organisms that have a nucleus. Okay, so we've got your diploid parent cell that you're starting out with, You're going to end up with 2 haploid cells that are genetically different from one another after meiosis I. And again, you're going to end up after meiosis II, you're going to end up with 4 genetically different haploid cells. So just to review, you need to know the difference between a diploid and a haploid cell in terms of chromosome number. You also need to know what cells in the human body are diploid and what cells in the human body are haploid. Okay. You also need to know what type of cell does meiosis begin with and what types of cells does meiosis I end with and meiosis II end with. How does meiosis ensure genetic variation among gametes? And then something to consider, why do we want to cut the chromosome number in half? So that's something that you should answer or at least think about so that we can talk about it during class. Hope you got down all the notes, and we will discuss more about meiosis in class, and then our next topic will be mitosis.